Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello, welcome to class. In the previous class, we have started with the discussion of blood and its composition, and we have discussed the first component of blood in detail, that is the plasma. And from this class, let us discuss the formed elements which are present in the blood. The first very important formed element found in the blood are RBC. Otherwise, they are called erythrocytes. RBC refers to red blood corpuscles. And the erythrocytes are the most abundant formed elements present in the blood. And for your imagination, on an average, normally there is presence of 5 to 5.5 million RBCs are present in a cubic millimeter of blood. That means in 1 cubic millimeter we could find the presence of 5 to 5.5 million number of RBCs. Just imagine the abundancy. And if the number decreases than this, if the number of RBCs decreases, this is the normal count of RBC. In a healthy individual, the normal count of RBC will be this. If the number decreases, then the condition is called anemia. The person is said to be anemic. It may be caused because of heavy loss of blood during the injuries or less production of RBC in the body itself. So that may cause the anemic condition. And as the name suggests, RBC, red blood corpuscles, they are red in color. They are red colored. And this red color is because of the presence of a red colored pigment called hemoglobin. Hemoglobin is the red colored pigment responsible for the red color of RBCs, and this hemoglobin is made up of, it is actually a protein, conjugate protein. Hemoglobin is a conjugate protein, complex protein, conjugate protein made up of protein part and a non-protein part. That's why we are referring the term conjugate. So here it is made up of, hemoglobin is made up of protein part and non-protein part. The protein part of hemoglobin is called globin. Globin is the protein part and the non-protein part is called heme part. And the hemoglobin is capable of forming a readily dissociable complex with oxygen to form a complex called oxyhemoglobin. That means hemoglobin can react readily with oxygen and form a dissociable complex called oxyhemoglobin. Remember, any complex which is formed in our body related to blood, they must be readily dissociable. They should not be firm. So with the change in the environment, they should change, because oxyhemoglobin is not dissociable complex. If it is a non-dissociable complex, then it cannot release the oxygen to tissue and it cannot take the carbon dioxide from the tissue. That's why it should form a dissociable complex. And so this oxyhemoglobin is capable of carrying the oxygen to each and every part or each and every cell of our body. Next, in our body, there should be presence of 12 to 16 gram of hemoglobin per 100 ml of blood. In every 100 ml, there should be presence of 12 to 16. The normal is found to be 14 in particular, but it ranges. So 12 to 16 gram of hemoglobin is compulsory and it is normal. The normal amount of hemoglobin present in blood. Next, the RBCs in case of vertebrates. In case of vertebrates like amphibians, reptiles, birds, the RBCs are having oval shape, oval and biconcave, oval, biconcave in shape, and they have the nucleus. So we refer the term nucleated. In case of vertebrates like amphibians, reptiles and birds, the RBCs are oval, biconcave and they are nucleated. That means they have the nucleus. Whereas in case of mammals, the RBCs are circular, biconcave and denucleated. Mammals do not have the nucleus. That is, the RBCs of mammals are devoid of nucleus, but there are exceptions, except in case of camel and llama. In camel and llama, the nucleus is present in the RBCs. Apart from this, all the mammals have circular, biconcave, denucleated RBCs. And if we look at the structure of RBCs, I told it is circular when we see it from the front. When we see it from the side like this, biconcave shape can be observed like this. The ridges are thicker than the rim. So here at the central part there is a thinner part. So the edges are thicker, the center part is thinner. This is because here this shape will increases the surface area. Increases the surface area. For the function of RBC is to carry the oxygen, right? So for increased supply of oxygen to the tissues, the RBCs are having the special shape. It is the circular biconcave shape. That is, the thinner inner part, that is the middle part, will support the increase in surface area, so that 20 to 30% increase in the supply of oxygen. So this is shape, only this is shape. That is, hemoglobin will carry the oxygen, that is the first part. But this shape will increases the supply or the formation of oxyhemoglobin by 20 to 30%. It may be asked in the competitive exams. The biconcave shape, why it is significant? Because it increases the surface area for the supply of oxygen to tissues by 20 to 30%. So that is the general thing we should know about the RBCs. During exercise and heavy work, there is a increase in the production of or the number of RBCs. Number of RBCs will increases. There is an increase in number of RBCs during the exercise, because during exercise there is a heavy burnt of nutrient that is in the presence of oxygen. For that, the cells need more amount of oxygen that should be supplied from the RBCs. So for that, our body will start producing more number of RBCs. And also in high altitudes, to meet the demand of oxygen by body, that is, in high altitudes the oxygen concentration in atmosphere will be less, but our body needs sufficient amount of oxygen. To supply that, our body will start producing RBCs. That is in two conditions, in high altitudes and during the exercise. That is a normal situation. If the number of RBCs are produced in more number in high altitudes and during exercise, that is a normal case. But if there is a abnormal production of RBCs, the abnormal increase in RBCs, even we are at rest and we are not in a high altitude, in a normal condition, if we are staying in a normal condition, then also our body start producing more number of RBCs. Then the condition is called polycythemia. So this is a clinical condition. And one more term is there. If there is decrease in number of RBCs, the condition is called erythrocytopenia. So these two terms you should remember. Polycythemia, it is abnormal increase in the number of RBCs, and if there is a decrease in number of RBCs, then the condition is called erythrocytopenia. If in our body there is a decrease in number of RBCs, decreased number of RBCs, then this condition will be detected by, the signals will be sent to the kidney. The kidney cells get stimulated. That means if there is a decrease in number of RBCs, the condition should not be like that. In our body, all the systems are like that. They are capable of repairing on their own. So here, if the number of RBCs decreased, then that situation will be red, or it is detected by the cells of kidney, and the cells, kidney cells will produce a hormone called erythropoietin. Erythropoietin hormone will get produced, and this erythropoietin hormone stimulates red bone marrow to produce RBC. So that means RBCs are produced in red bone marrow, that is in adults, that is in our body. The decreased number of RBCs are there. Don't think only during decreased condition. Normally there is production of RBCs. taking place. But if in under certain circumstances the count decreases more than the normal, then this process will take place. Erythropoietin hormone will get produced from the kidney. It will stimulate the red bone marrow to produce the RBC. And the production of RBCs is called, by the process called production of RBCs. The process, the formation of RBCs is called, the process is called erythropoiesis. Erythropoiesis. And for this process, our body required special nutrients like vitamin B12, folate, that is folic acid, and very importantly, iron. Iron is required. So, for erythropoiesis process. The erythropoiesis process in case of adults takes place in red bone marrow, as I told, and in case of early embryonic stages. In early embryonic stage, the erythropoiesis process takes place in kidney and liver. So these terms you should remember. In adults, it takes place in red bone marrow. In case of early embryonic stages, it takes place in kidney and liver. And this erythropoiesis process takes place for about 72 hours. Erythropoiesis process will get completes in 72 hours. And after the formation of RBC, it will be functional, that is, it is transport carbon dioxide and oxygen, that is, from the lungs it will carry large amount of oxygen, and from the tissues it carries some amount of carbon dioxide, not huge amount. And this is called normal functional RBC. After 120 days, the RBCs will become non-functional. That is, the normal lifespan of RBC is 120 days. Normal lifespan of RBCs is 120 days. That is, after that they will become non-functional. After 120 days, the RBCs will get destroyed by phagocytosis. Destroyed by phagocytosis in spleen. The spleen cell will phagocytosize the RBCs, and hence spleen is also called graveyard of RBC. So you may ask with the question, why spleen is called graveyard of RBC? Because after 120 days of lifespan, the RBC cells are phagocytosized in the spleen, and they will be converted or they will be dissociated into pigment and the iron. So the iron will be utilized for the formation of fresh RBCs in the bone marrow, and the remaining things, that is the pigments, will get converted into the coloring pigments like bilirubin and biliverdin. They are converted into bilirubin and biliverdin. So if the amount of these two increases in our body, then they get, if they are not get excreted completely, then they start depositing in mucous membrane and in the skin, and condition we know it leads to jaundice. So this is about the detailed thing we should know about the RBCs, and you remember the things like the erythropoiesis and nutrients required for erythropoiesis, polycythemia, erythrocytopenia, then graveyard of RBC. These are the key terms you should remember regarding the RBCs. And from the next class, we shall continue with the leukocytes and the thrombocytes in detail.